All right, so welcome to our group presentation for wildlife behavior. My name is Taylor. My group mates are Jordan, Kirsten, and Emily, and we worked on a study into the activity budgets of wintering waterfowl in the Atlantic Flyway. So I'm going to give everyone a bit of background on the subject. Then Emily will go into the methods of our study, Kirsten will discuss our results, and Jordan will discuss our conclusions. So first off, if you aren't too familiar with the Atlantic Flyway, it's one of the 4 flyways in North America. It's along the east coast of the US. You can see it shaded in purple there, from up in Connecticut all the way down to Florida. And it's an important migration route for a plethora of different species. But we are particularly concerned with waterfowl for this study. And here are some of the specific species that we were able to look at. So starting from the top left and working our way down, we have the northern pintail, then below the northern shoveler, the American widgeon, the tundra swan, the Canada goose, the hooded merganser, the wood duck, the bufflehead, the gadwall, the American black duck, the ruddy duck, the ring-necked duck, and finally the mallard. So like I said, our study is related to the activity budget of these birds. An activity budget is essentially the amount of time an individual spends in different behaviors. So it can be really helpful to quantify the behavior of a specific species. They're typically presented as the proportion or percentage of time that the animal spends on that particular activity. And they're important for providing like baseline data on animal behavior. They can be used for testing various hypotheses related to different experimental treatments or perhaps to measure response to an environmental change. In order to conduct an activity budget, it's really important to divide the animal's behavioral repertoire into mutually exclusive categories. So if behaviors aren't clearly defined and understood by all the observers, it can lead to individual error or bias in the results, especially in a project like ours, where group members are working in different regions. So we first had to decide what behaviors we would focus on and how to recognize and define each behavior exclusively. And Emily will go into the specifics of how we did that in our methods section. But then we had to decide whether we were going to use continuous or instantaneous data. As we've learned in class, continuous data refers to observational data collected in a continuum over a predetermined time period. The percent of time spent in that activity, in each activity, in this case, would be the duration of time in that activity divided by the total sampling period. So let's say you record for 5 minutes total continuously, you would then look at what percentage of that total 5 minutes they were doing a particular activity. However, we chose to use instantaneous data, which of course is also observational, but it's collected at an instant during specific intervals over a period of time. So in that case, the percentage of time in each activity would be the number of instantaneous recordings of each behavior divided by the total number of instantaneous recordings of all behaviors. And we felt this would be a better approach to our study. And so we decided on instantaneous sampling, instantaneous focal sampling specifically. So that means that we would be observing an individual bird for a specific period of time. In our case, it was 5 minutes, and then recording our instantaneous data, and then moving on to another bird rather than looking at the entire flock of birds and recording the data for all the individuals. So by using focal instantaneous data, we're able to account for individual variation among focal birds in our statistics. So now that everyone has an idea of what kind of study we conducted, I'll pass it on to Emily to discuss just how we conducted it in our methods.
So our hypothesis for the study was that the average time spent feeding between observed waterfowl species will differ because of species-specific dietary differences. So as you can see in this video here, there's some blue-winged teal and northern shovelers dabbling on the surface for invertebrates. So we hypothesized that the dabblers would spend more time feeding than the divers because divers are able to go deeper under the water to obtain more energy-rich and nutritious foods than the dabblers are. So our objectives for this study included 1, to separate waterfowl behaviors into mutually exclusive categories, 2, examine any possible differences in feeding activities, 3, determine the percent time feeding in each species sampled. And then finally, 4, determine any statistical significance between the time spent feeding in observed species. So these were our 4 study sites, and we were located in 3 states within the Atlantic Flyway. So I was located at Matamuskeet National Wildlife Refuge in North Carolina. Jordan was located in Wilmington, North Carolina. Kirsten was at the Bombay Hook National Wildlife Refuge in Delaware. And then Taylor was at the Powwow River in Massachusetts. So to conduct our activity budgets, we selected a single individual, observed that individual for 5 consecutive minutes, and then recorded their behavior every 10 seconds. So this would give us a total of 30 recorded observations for a single individual. So each of us attempted to collect data on a total of 10 individuals within 5 species. So that would give us, between the 4 of us, a total of 200 individuals. Data was collected every Monday, Wednesday, and Friday from 8 to 11 a.m., just to keep days and times consistent between all of our states and sites. So this is a quick look at our activity budgets data sheets. So we had a general folder, activity budgets folder, and within that folder, we all had our own personal folders. And within our own personal folders were 5 tabs, where each tab corresponded to data for a single species. So the first section of data that we would collect would be, date, time, observer, temperature, which was broken down into brackets or categories for analysis, and then same thing with cloud cover, precipitation, and habitat. This lower section here is where we would actually enter our observations. So as you can see, these are my recordings for, 5 of my recordings for American widgeon. So if you look at the first male, the second individual observed, there's only a total of 28 observations because that particular individual was out of view 2 of the times. So for 20 seconds, that individual was considered to be out of view. And when you're analyzing the data, you don't include the out of view observations for the species. So as an overview of how we broke down our behavior categories, we considered feeding to be behaviors associated with food or water intake. So this included dabbling, which you saw in the first video, tipping up, which you see here in this picture, and then also slow swimming while dabbling. And then, as I mentioned earlier, divers feed underwater, so the bird is considered to be feeding while it's underwater, and then feeding ceases once it resurfaces. So the alert behavior was attentive postures that usually interrupt other activities. So for example, if a bird is preening and it suddenly stops, like twitches to the left, looks up, that's considered, and then resumes preening, that's considered to be an alert behavior. And then also in this picture here, you can see the male wood duck, his neck is stretched out. That's also considered to be an alert posture. Birds in general rarely conduct activities for any period of time without frequent nervous or alert interruptions. Next was resting. So you can see here in this picture of the 2 blue-winged teal. Resting, we can define resting as static postures associated with loafing and sleeping. So birds show a relaxed posture when they're resting their head on their shoulders. So that's kind of like the no neck look. And then also when their bill is tucked under their wing, as you can see with those teal there. Comfort behaviors were those associated with body maintenance, such as preening, stretching, shaking feathers, bathing, and then also tucking one's legs up towards its breast. Social interactions were defined as courtship or aggressive behaviors directed at 2 or more individuals. So as you can see here in this little video, there's a mandarin duck kind of going after either a ringneck duck or a tufted duck, it looks like. But for social interactions, one bird is the initiator and then the other bird is the receiver of that interaction. Our last behavior we defined was locomotion. So that was spatial movements such as flying, swimming, and walking, which is independent of other activities. And then finally, just in general, was the out of view recordings. So that's when the bird has flown off and we can't see it anymore, or that's when the bird is out of view because it's behind vegetation.
Over the course of our study period, we observed a total of 178 individuals that belong to 13 different species. You can see the distribution of those species in this pie chart over here. So that would be a total of 20 northern pintails, 20 northern shovelers, 34 mallards, 10 American widgeons, 10 gadwalls, 10 American black ducks, 8 wood ducks, 10 buffleheads, 14 hooded mergansers, 10 ruddy ducks, 2 ringneck ducks, 20 tundra swans, and 10 Canada geese. And we can split up those species into 3 categories. So the first category would be dabblers, those species of ducks that spend their time paddling on top of the water searching for food. Those include the pintails, the shovelers, the mallards, widgeons, gadwalls, black ducks, and wood ducks. So this here is our raw data, the number of instances that we observed the individual conducting that specific behavior. But if we turn that into a percentage, we can see that the dabblers spend a total of 41% of the time feeding, 19% of the time resting, 20% of the time in locomotion activities, 7% of the time alert, 9% doing comfort activities, and 3% in social activities. And then the next group is the divers, and these are the ducks that'll dive underwater foraging for food, and these include the buffleheads, hooded mergansers, ruddy ducks, and ringneck ducks. Again, this is our raw data here that will convert into a percentage, and we'll see that the divers spend a total of 47% of the time feeding, 7% of the time resting, 36% of the time in locomotion behavior activities, 5% of the time alert, 5% of the time in comfort activities, and less than 1% of the time doing social activities. And then our third and final group is our swans and geese, which are just made up of the 2, the tundra swan and the Canada goose. And our raw data here, we will convert into a percentage and find that they spent a total of 22% of the time feeding, 35% of the time resting, 8% of the time in locomotion activities, 13% of the time alert, 16% of the time in comfort, and 5% of the time in social activities. And here is just a visual of all of those 3 different groups together in a chart form, and you'll see that the blue is the dabblers, the orange is the divers, and then the gray is the geese and swans. So that shows you how much time respective of each other that they spent in each behavioral group or behavioral activity. So to find the variation within the groups, we used a single factor ANOVA analysis, or analysis of variance. So for the dabblers, this was our p-value, and because of this, we're rejecting the null hypothesis because the p-value is less than 0.05. And therefore, our alternative hypothesis is that there is a statistical difference in the calculated percent of time spent in observed behaviors within the group / species. And then for the divers, we did the single factor ANOVA again. This is our p-value, and here we again are rejecting the null hypothesis because the p-value is less than 0.05%. So alternative hypothesis again is that there is a statistical difference in the calculated percent time spent in observed behaviors within the group species. And then finally for the geese swans, we got a p-value that was above 0.05, so that leads us to fail to reject the null hypothesis, which would be that there is no statistical difference in the calculated percent time spent in observed behaviors within the group species. And then the variation between groups, we just looked at the time spent feeding only. We used that single factor ANOVA again. Our p-value is shown, and that means we're failing to reject the null hypothesis because the p-value is greater than 0.05. So we stick with the null hypothesis, which is that there is no statistical difference in the calculated percent time spent in observed behaviors. That's supposed to be between groups. This is between all three of the groups.
Hey, thanks, Kirsten. I'm Jordan Brookshire, and we'll just talk about the conclusions real quick. We'll talk a little bit about our hypothesis and whether we support or reject that hypothesis. We'll talk about how we met our objectives, some obstacles and limiting factors that we came across, and then some further research that could be done or different aspects that could be done on a larger scale. So let's talk about our hypothesis real quick. So our hypothesis, as Emily pointed out, was that average time spent feeding between observed waterfowl species will be different because of the species' specific dietary needs and differences. So we also hypothesized that dabblers would be spending more time feeding because divers can go deeper during their dives to obtain more nutritionally dense foods. And then, so breaking that down, kind of reiterating what Kirsten said, is that there was a significant difference between the groups or between the species within each group, whether it was our dabblers or our divers. There wasn't a statistical difference between the geese and swans group, but that also can be attributed to such a small study group that we had for them. But then there, in between all of these groups combined, the three groups, there wasn't a statistical difference in the time spent in the observed behavior. So, so as you'll see in our next slide, our dabblers really didn't time than our divers feeding. So we'll get to that. Our objectives, as Emily stated, were to separate the behaviors into different categories, and we did that by changing them into feeding, alert, resting, comfort, locomotion, and social. And then we examined the differences in the feeding strategies, and all of us learned something very in particular about each of these species and how they feed. And it was awesome to just sit there and watch different species dabbling or diving or just foraging and interacting with each other. So it was great for all of us. We all enjoyed it. And then determining the percentage of time feeding by species, we did that, as you've seen the raw data. And then determining that statistical difference between the groups, as we touched on just a second ago. And the raw data, to reiterate, the dabblers spent 41% of their time, divers 47%. So that kind of goes back to our hypothesis of, we thought that dabblers would be spending more time foraging, but that wasn't the case. So some limiting factors that we came across, a big one was weather. There was a lot of inclement weather during our study period that kept either the waterfowl hunkered down in the areas that they were nesting or not foraging, or it was just impossible to get out to the study sites. Flyway arrangement had some factors to do with this. As you noticed earlier, we're all very coastal, all on the coast. So seasonally, the birds could have been pushed due to some of that inclement weather or the changes in the weather. Some days are super cold, some days it got really warm during our period. So they moved rapidly at certain times or stayed rapidly. So that kind of impacted our species richness that we got. And also just being on the coast over here, we're a very small strip of the flyway. So if we were out in Western North Carolina, so for me and Emily, then we would see a completely different array of species and probably have a larger species richness, excuse me. So some further research that could be conducted for this, is to spread it out. We had a very large study area all the way up, into the northern states. And so that Atlantic Flyway goes a long way. So we could extend this out and have way more observations, a longer time. And then there could be some specifics about like habitat, food resources that are being predated on primarily. We as a group looked at our habitats and annotated that, but there could be a lot more done with that, whether it's depth, time, temperature, all these different aspects could be put into play to get some more specific information on these species. So with that, thank you very much for watching. I hope you really enjoyed it. We had a great time doing the study, and thank you to all of my fellow cohorts. We we enjoyed it. Thank you.